I'm Dr. Marilyn Yoder. My research interest is in structural biology, which means I use high-resolution atomic level structures to try to explain the function of biological molecules. When I first saw this picture of the SARS-CoV-2 structure, I thought it might be based on experimental X-ray diffraction or electron microscopy data. It is not, but it is based on experimentally determined structures of components of the virus, as well as our understanding of the original SARS and the MERS viruses. So I'm going to walk you through this particular structure that was created by the Centers for Disease Control and Prevention. In this picture, the gray area represents the phospholipids that form a membrane that we call the envelope. Most animal viruses are enveloped. They are surrounded by a membrane similar to human cells, which are also surrounded by a membrane. In fact, in SARS-CoV-2, it is a human membrane that forms this envelope. All of the components in the virus are manufactured in the host cell's machinery. With COVID-19, the host cell is the infected human. The membrane is composed of phospholipids, which is a fat molecule that is synthesized by the human host. One function of a membrane is to form a barrier or a boundary. So I'm going to elaborate a bit on the envelope structure for reasons you'll see towards the end. This is a blow-up of a bilayer membrane. It has two layers, one on the top and one on the bottom. And it has a hydrophobic core in the middle. And hydrophobic means that it is an area with a very strong aversion to water. So you will find water on both sides of the membrane, on the top here and the bottom, but you will not find water in the middle of this hydrophobic core. So the other major part of the virus that's seen in this image are these red triangles, and they are spike proteins. It is a viral protein that is inserted into the envelope. So part of the protein extends into the membrane, and we cannot see it, or inside the envelope, and the other part extends outward from the exterior of the virus on the surface. In SARS-CoV-2 and other animal viruses, these spike proteins bind to receptors on the host cell. And again, the host cell is the human cell that has been infected. The receptor is on the surface of the human cell, and it is able to specifically bind to spike proteins, which are on the surface of the virus. And this is the first step in the virus entering the cell. If there is not a receptor on a host cell, the virus cannot bind, and there is not entry of the virus into the cell. If one is trying to design pharmaceuticals or antibodies, knowing the receptor that the virus binds to is really significant. We now know that the spike protein of SARS-CoV-2 binds to human angiotensin converting enzyme, which we call ACE2. And so ACE2 is the receptor for SARS-CoV-2. ACE2 is also the receptor for the original SARS virus, but MERS, which is a different coronavirus, has a different receptor. The structure of the virus also explains why washing your hands with soapy water is a helpful deterrent for infection. Soap actually has a similar structural features to the phospholipids in the envelope of the virus. So I'm going to show a clip of a video created by the Protein Data Bank, which is a consortium of research institutes that house structural data. And in this video, we have a change in our color scheme. So the coral bumps that you see down here represent the membrane and the envelopes. It was gray in the CDC image. The green teal here is the spike proteins, which was red in the CDC image. And these lime green things are the soap molecules. And we will see how the soap molecules interact.
The lipid ends of the soap molecules are attracted to the lipids in the membrane. They take advantage of the presence of membrane proteins, which can perturb the neatly organized bilayer, to insert themselves into the viral membrane. If there is only a small amount of soap, the soap molecules only loosen the membrane. But with more soap, they begin to create micelles around membrane lipids. They are also attracted to hydrophobic amino acids in the membrane proteins, extracting them from the membrane. The structural integrity of a virus is essential for infection.
Therefore, washing your hands with water will not disrupt the virus structure. If you're lucky, you'll rinse the virus away. But soap has two advantages. One, it forms bubbles and suds, so you know where on your hands the soap is, so it gives you a good indication of how well you are soaping your hands. Secondly, the soap breaks the virus apart. Viruses are not living organisms. They need to be destroyed. So they can be destroyed by soap, as shown here, or they can be destroyed by antibodies and your immune system. So I hope that this video has explained some aspects of the structure of the SARS-CoV-2 virus that is shown in this image that we see frequently in news and multimedia sites.